Thanks a lot. This is an absolute pleasure to be here, and I would like to thank the organizers for this invitation. So I'm going to present our main goal in the lab, which is trying to understand the regulation of lipid metabolism in neuronal biology. And especially with a special emphasis on trying to understand the cause and relevance of the many lipid alterations that we can see in neurodegenerative diseases such as Alzheimer's, PD, and ALS, as well as other metabolic insults such as mitochondria. So why lipids? I know that everybody hates lipids, but let me tell you that lipids are, of course, the main components of membranes and the determinants of membrane biology. It is believed that more than 4,000 lipids exist in mammal cells. Why all these many lipids? Because every combination of a lipid of these lipid species is going to actually give rise to all the different cell types. And the structure of these cells, every single structure is absolutely essential for the functioning of the cell and the regulation of the cell. So the more complicated it is the shape of a cell, the more it relies on its lipid composition and the regulation of lipid metabolism. Similarly, actually, every single intracellular organelle has a specific lipid composition in order that allows them to perform their specific functions. The lipid composition of a cell and an organelle is a characteristic acquired during development, and it's a constant. So the cell has to maintain it constantly in order to do its functions. There is a misconception in the field, which is lipids do not have activity. They do not have enzymatic activity, of course, but they do have biophysical activity in response to changes in the environment. So any kind of enzymatic activity, it has to be followed by changes in lipid metabolism and vice versa. So any kind of change, biophysical change, can actually alter, if you change the lipid composition of the membrane, it's going to alter the conformation and the distribution of a specific membrane proteins, as well, of course, as it's going to help the oligomerization, the interaction between different proteins, as well as regulating the curvature of the membrane in processes such as vesicle formation or synaptic transmission. Also, the lipid composition of a membrane regulates the permeability of the membrane and specific ionic channels, as for instance, the lipid composition of a membrane regulates the activity of potassium channels during action potentials. Two of the main cells that rely mostly, the cells that rely the most on their lipid composition and the regulations are neurons and macrophages. Neurons, of course, because there is a constant turnover of lipids in order to sustain synaptic activity. But in macrophages, it's already well known that the activation of different type of macrophages depends on the lipid raft composition, meaning the lipid composition of the membranes, and also specific signatures or signaling mechanisms such as TLR4, for instance, is extremely dependent on cholesterol and sphingolipid regulation. In the lab, actually, if we think about, so the lipid composition of a cell is a characteristic acquired during development. So how is this lipid composition maintained in the cell is one of the questions that we have in the lab. In a paper, quite recent actually, one group has demonstrated that the regulation of lipids is not linear, it's circular. What it means is that a change in one specific lipid species is going to be mirrored by another change in order to sustain homeostasis. So this means that all lipids are interdependent, meaning you have to analyze lipid biology and lipidomics as a whole, not as individual species. So the other thing that we just have found is that although all lipids are interconnected, not all are equally important, meaning there is a hierarchy in lipids. Hierarchy based on hydrophobicity. The more hydrophobic a lipid is, the less the cell can tolerate changes in that specific lipid. While other lipids that are less hydrophobic can be changed, those changes or alterations in the concentration of those lipids are better tolerated by the cell. So based on hydrophobicity, the king of lipids is cholesterol. In our cells, in mammal cells, cholesterol is the most hydrophobic lipid that you can find, which means that very little changes in cholesterol that are non-statistically significant can be extremely significant in terms of the biology. So, because, probably, any change that more of 0.1% of cholesterol can trigger cell pathology. As, you well know, most of cholesterol is believed to be synthesized de novo in the endoplasmic reticulum. Here, there is a very complex pathway to synthesize cholesterol. This is the enzyme that is the best known, the HMG-CoA reductase, which is the target of many statins. However, the synthesis of cholesterol is a very complex series of different enzymatic reactions, some of which are actually also rate-limiting as HMG-CoA reductase. Also, this pathway, we have to take into account that regulates many other functions, such as CoQ synthesis, farnesylation, or even prenylation. So if cholesterol is not synthesized, it can also be uptaken from the blood, from the environment. Many cells rely on the uptake of lipoproteins to fulfill their needs for cholesterol. What is really well known, thanks to the work of Nobel Prizes, Brown and Goldstein, is that when cholesterol is in excess, the plasma membrane invaginates and sends that excess of cholesterol towards the endoplasmic reticulum. That excess of cholesterol arrives to the ER, and as described by these two investigators, in a manner that when cholesterol is high in the ER, when it arrives, it's going to inhibit the de novo synthesis. On the other hand, if cholesterol is low in the ER, it's going to stimulate cholesterol synthesis. So it seems that there is a very interesting intracellular pool of cholesterol in the endoplasmic reticulum that seems to regulate the equilibrium between the synthesis of cholesterol and the uptake of cholesterol. So we decided to focus on this interesting cholesterol pool that has received many, many names during in the literature. For instance, it has been shown to intracellular cholesterol pool of cholesterol in the ER, or even just cholesterol platforms in the endoplasmic reticulum. Our data from the last years actually agrees with this proposition. We have found that when cholesterol is in excess, indeed, it invaginates the plasma membrane, and cholesterol, the excess of cholesterol, is going to be shuttled to the endoplasmic reticulum as previously described. What we have found is that it's not homogeneously distributed. The interesting thing is that the cholesterol that arises from the plasma membrane forms a lipid raft, a platform in the endoplasmic reticulum that is in close association to mitochondria. This domain has been named mitochondria-associated membranes, or MEM. MEM is a lipid raft. What does it mean? It means that when cholesterol, because it's highly hydrophobic, if cholesterol increases, it's going to try to associate with other molecules of cholesterol, as well as many cholesterol binding proteins, in order to protect it from water. At the end of the day, what happens is that there is a formation of a cholesterol-rich domain that is also rich in a lot of different cholesterol-binding proteins. Now these proteins can actually interact with each other, can change different conformation. That's why we believe that this is a transient and a functional platform in the cell. Not only that, now, after several years where researching MEM has exploded, we know that this MEM, this platform, is enriched in several proteins that regulate lipid metabolism, calcium transference between ER and mitochondria, inflammatory reactions, mitochondrial biology, autophagy, and many others. Another thing, one interesting result that we just got is in the lab is we use cell models where we can induce the formation of the MAM or we can inhibit the formation of the MAM by manipulating cholesterol internalization. Interestingly, when we do that and we isolated the MAM and analyzed it by proteomics, we found that when MAM is activated, there is an enrichment in a lot of different enzymes that, as you can see, they're highly related to cholesterol turnover and cholesterol synthesis. Indeed, we have found that the MAM, when it's formed, recruits mostly all post-squalene enzymes, and not only that, it inhibits these enzymes. How? We found that the activation of MAM induces cholesterol internalization. Here you can see it in green. We add exogenous cholesterol, and in blue you can see the endogenous cholesterol. This is alpha-lipid staining. And we have found that when MAM is activated, we see a high increase in cholesterol uptake that is concomitant to a reduction in cholesterol synthesis in order to balance cholesterol levels in the cell. And also it affects HMG-CoA expression. On the other hand, when we inhibit the MAM, as you can see, exogenous cholesterol is now internalized, stays in the plasma membrane, and cholesterol uptake is reduced, as well as a concomitant increase in the synthesis of the novel cholesterol to balance the levels. So we repeated this not only with fluorescent cholesterol, we have repeated this with also radiolabeled cholesterol and some other precursors, and we have replicated this data. The conclusion is that this little domain in the endoplasmic reticulum participates or contributes to the regulation of the balance between the synthesis and the uptake of cholesterol. And of course, when we started thinking about cholesterol, we tried to apply what we have found to neurodegenerative diseases. The reason behind this is that the neurons, and specifically hippocampal neurons, are highly dependent on cholesterol regulation. And of course, as you well know, there is a lot of literature analyzing the progression of Alzheimer's, and they have seen, many authors have seen that lipid alterations are actually quite early in the disease and even before any kind of symptomatology in the patient. Moreover, cholesterol has been implicated in mostly any kind of symptom in Alzheimer's disease. As you well know, cholesterol has been found to regulate APP cleavage, has been implicated in inflammatory reaction, and many others. And in the last years also, we have seen an explosion of different genes and gene variants that related to lipids and cholesterol that are associated with a higher risk of Alzheimer's. Interestingly, years ago, in every single model of Alzheimer's disease that we could get, actually, we found that MAM, the connection between the mitochondria and endoplasmic reticulum, in here the ER is in green, and in red you can see the mitochondria. The interaction between the endoplasmic reticulum and the ER is quite high, or higher, in any kind of cell model of AD. I'm saying any kind of cell model because, of course, the reviewer told us to replicate this in every single cell, sporadic, familial, everything. So we are quite sure that this is the case. But not only that, the important thing is that the functions that are localized at the MAM are also regulated. For instance, in here we have there are two kind of accepted assays that are well known to measure MAM activity, one of which is the synthesis of phosphatidylserine at the MAM. It only occurs at the MAM and is transferred to mitochondria to synthesize phosphatidylethanolamine. So we can label substrates like serine and track the incorporation of the radiolabel into the different phospholipid species. So when we measure this, we found that in that AD cells have a higher rate of phospholipid synthesis and transfer, indicating that indeed there is an increase in MAM activity. Not only that, we also, another assay that is a well-accepted measure of MAM activity is cholesterol esterification by the HCl cholesterol acetyltransferase 1. This enzyme, ACAT1, esterifies excess of cholesterol into cholesterol esters, and those cholesterol esters are going to be stored into lipid droplets. The activity of this enzyme, again, is highly upregulated in cell models of AD as well as in tissues. But not only that, in support of that, we have found that Alzheimer's cells, familial, sporadic cell models, there is a significant increase of lipid droplets in the cytosol. So then we decided to understand what is the reason why MAM is upregulated in here. So in order, we try several approaches, but I'm going to show you the one that works, of course. And that is, we started trying to understand what is the relationship between MAM activation and APP cleavage, if any. So for that, we purified brain homogenates through sucrose gradients, and then analyzed the different fractions of the sucrose gradient by Western blot using specific markers for different compartments. For instance, we use VDAG1, that, as you all know, is a marker of outer membrane of the mitochondria, but we know that VDAG, although it's in the outer membrane of the mitochondria, is quite enriched in those areas of the mitochondria that are in touch with the endoplasmic reticulum. Similarly, we found that SACL4, which is a very well-known MAM marker, this protein only localizes to MAM and is only active at the MAM, was also enriched in this area of the gradient. Similarly, and as previously shown, we found that presenilin-1 is also enriched in those areas. We used several other markers, and we just ended up defining an area in the gradient that we know is enriching MAM markers. And to our surprise, APP full length was not even close to be localized at the MAM, nor actually were the C-terminal fragments of APP, which are actually the substrates of the gamma secretase. So this kind of mislocalization between the gamma secretase catalytic core and the substrate of the gamma secretase has been coined in the field as a spatial paradox. So indeed, the carboxy terminal fragments of APP co-localize with endosomal markers as previously shown. So but then reading a lot about the biochemistry of gamma secretase, it seems that the cleavage of presenilins, it's so fast that it's impossible to all detect. It's extremely fast. So we decided to repeat this same analysis by inhibiting the gamma secretase. And now, to our surprise, we found that there is a certain amount of C99, the C-terminal fragment that is the precursor of amyloid, enriched in those areas that co-migrated with MAM markers. So we decided to start focusing a little bit on C99. What can be the reason why this enzyme, with this peptide can increase MAM formation? Indeed, we found that when we inhibit the gamma secretase, or when we transfect C99 plasmids here that are labeled with a red fluorophore, and together with a marker for the endoplasmic reticulum, mitochondrial marker, in the merge we found that C99 accumulates in areas of the endoplasmic reticulum that are in close contact to mitochondria. And similarly, in some Alzheimer's models, we found that by EM, we found an accumulation of this fragment in areas of the endoplasmic reticulum that are in close opposition. Let me remind you that, of course, A beta is the main character in Alzheimer's disease. It's not a new concept that C99 actually correlates very nicely with the cognitive impairment. It accumulates in the disease as has been shown several times, and it actually, associates with the progression of disease in a matter closer to tau and in a much better way than A beta. So, indeed, we found that this accumulation of C99 in the endoplasmic reticulum induces the formation of MAM domains and induces also the uptake of extracellular cholesterol as well as inhibiting cholesterol de novo synthesis. Again, so we found that in cells, sporadic AD, familiar AD, as well as in many models, there is a clear increase in C99 levels, independently of A beta. And this increase in C99 was associated with an uptake, a significant increase in the uptake of extracellular cholesterol, here as you can see labeled by Lipid, that we can also quantify this by radioactivity using different substrates. And this increase in the uptake due to the accumulation of C99 also was associated with an increase in MAM activity and a decrease in the de novo synthesis of cholesterol. So again, in order, trying to understand what's going on, we decided to repeat these experiments in the presence of a BACE inhibitor to impede the formation of the C99. And we can rescue those phenotypes now. When we expose Alzheimer's cell models to BACE inhibitors, we find that there is a decrease in the internalization of cholesterol as well as a reduction in MAM activity. So the question that you should ask me now is that, so why BACE inhibitors didn't work in the disease as a therapy? Why? What is the problem in here if it is such a good inhibitor? That actually led us to the other side of the lab, led mostly by the most talented postdoc that I have met, Delfina Larrea. What Delfina has found is that when MAM is formed, there is a recruitment of a lot of different glycolytic enzymes that allow for the formation of pyruvate and the entry of pyruvate inside the mitochondria in a manner, and to produce ATP, of course. So when Delfina inhibits the MAM, the formation of the MAM, we found that there is a decrease in pyruvate metabolism and a counterbalancing effect. So when mitochondria does not have access to pyruvate, it's going to use alternative substrates, such as fatty acids and amino acids. In using models, in this case, these are motor neurons derived from stem cells, with mutations in one of the main genes associated to amyotrophic lateral sclerosis, SOD1. When we analyze cholesterol internalization, in these cell models as well as in tissues from these patients or animal models, we found that there is a clear and significant decrease in cholesterol uptake and a counterbalancing mechanism, a buffering effect trying to increase the de novo synthesis of cholesterol. This, of course, is associated with a significant reduction in MAM activity. And not only that, what Delfina has found is indeed this alteration is associated with an impairment in pyruvate production and pyruvate use by the mitochondria that is buffered by an increase in the use of fatty acids as a substrate for ATP production. In principle, this will not be a big deal. But let me remind you that the oxidation of pyruvate can produce, and then entry into the TCA cycle, we can produce 34 ATP molecules. However, per fatty acid and beta oxidation, we can only produce 5 ATP. And not only that, 5 molecules of ATP, not only that, beta oxidation actually carries a lot of different, it initiates a lot of different signaling mechanisms that are going to end up in the higher production of superoxide and hydrogen peroxide in the cell. So as such, this is not a problem for many cells, but for cells that are highly demanding, such as motor neurons, and over time, in acute phase, fatty acid burning is not a problem, but over time, it's going to increase an accumulation of specific radicals as well as a bioenergetic crisis. And so what I would like to summarize in these works is that the cell models of AD present with an accumulation of C99 fragments that in the ER, by its affinity for cholesterol, induces the formation and the upregulation of MAM domains. The accumulation of C99 in MAM domains induces the uptake and internalization of cholesterol from the plasma membrane to the ER, maintaining the activation of MAM. Moreover, alterations in MAM result in the loss of lipid homeostasis and the regulation of lipid composition of cellular membranes in cell models of AD. And this last sentence is what we are using, trying to make our work a little bit more translational. So, as Carmen said, from bench to bed, what we have found is that the activation of MAM, the regulation of MAM, actually is the basis to regulate lipid metabolism in the cell. Many different enzymes are localized and activated at MAM domains. All these enzymes, for instance, that I mentioned before, are involved in the regulation of cholesterol, as well as a lot of enzymes involved in sphingolipid regulation, and as well as in phospholipid, fatty acid synthesis, etc. What happens then is that we have track the alterations in the lipid metabolism that occur when MAM is altered. And we have found that these alterations are not random. So there are specific lipid species that get affected by alterations in MAM that can be tracked over time. And I don't have time to show you all the data, but we've been working for the last 5 years on different samples in longitudinal samples, trying to understand how MAM is changing the lipid composition of different samples, such as serum, fibroblasts from patients, and PBMCs. For that, we're using actually not only sporadics are a little bit more difficult, but we are actually doing longitudinal studies in the biggest cohort right now of familial AD patients in Colombia. So using these samples, we're trying to analyze the alteration or the modification, the progression of these lipid alterations in serum from patients from 8 years to 40 years. And I'm quite hopeful that we are finding something, so I'll keep you posted. And of course, the most important slide. Here are the main characters involved in this work. Jorge Montesinos, which is the leader of the Alzheimer's project, together with Rishi Agrawal and a previous postdoc in the lab, Marta Vera. And Kirsten, our students, Delfina Larrea and Kushbu, which are the site in ALS. And of course, Kevin and Danny, which are always taking care of us. And of course, thank you very much to all our collaborators, a special mention to my partner in crime, Eric Sean and Carmen Paradas. And of course, our funding sources. And to you for your attention. Thank you very much.